Welcome everybody to our monthly webinar series, and we have a very interesting topic to discuss, which is that about understanding the requirements of Annex 1 when it comes to contamination control for the transportation of sterile components that are used in the process. So we want to dive deep into how we can reduce contamination, and I'd like to introduce Elena from AM Instruments. Elena is a specialist. Let me get onto her page. She's a specialist in pharmaceutical packaging solutions and has previous experience as an R&D engineer in the medical device industries. Welcome, Elena, to our webinar, and it's nice to have you here.
Thank you for the invitation.
You're welcome. And I'd like to say you have some good experience. I see R&D engineer in the medical device industries. Can you tell us a little bit about yourself there?
Yes, I work almost 5 years in two main industries in medical devices section, and I was an R&D engineer in both cases. In one case, I follow a study on dialysis project, and in the second case, I was in charge of projects related to monitoring system during cardiac surgeries. And so it was a big opportunity to see also operating rooms.
Yeah, that's interesting. My nephew actually works for a medical device company. He goes to the hospitals and observes the process during the actual medical procedure to validate that everything had been installed correctly with the heart, devices. So very interesting. So you're well aware of the production in a medical device facility and the need to control contamination. And as you move over into pharmaceutical industry, what differences have you seen between medical device and pharmaceutical industry?
For sure, pharmaceutical industries have more strictly requirements regarding, Annex 1 requirements are much more stricter than the requirements that we have in the medical devices, even though also in these fields they are becoming stricter. So this is the main difference, the use of material. The two processes are different, but I think that in particular, considering medical devices that goes in a deep contact with the patient, so with blood, bones, this kind of devices, will arrive to the same level of sterility that we have in the pharmaceutical field.
And I see you position yourself as a subject matter expert in the contamination control solutions. You mentioned Tyvek, the aseptic process. Obviously, the industries you cover, medical device, life sciences, even semiconductor. That's interesting.
Yeah, these all characteristics, these all experiences are thanks to AM because, of course, are all focused to the pharmaceutical industries and also semiconductor. A little bit for my previous experience because I worked with also at a hardware level. But also thanks to AM because we are providing the specific Tyvek solutions also in this sector to protect the buffer with chips from dust and preventing, the possibility to contaminate these critical items.
Interesting, because your experience does span across the whole clean room industry nearly. You look at all of the companies that you've or the industries that you've worked in, and your expertise from technical support in the field in Italy and Europe. You travel around to many customer sites, project development, the technical development of packaging solutions. You've covered many presentations in Italy, Europe on GMP packaging solutions, and you got a black belt, Six Sigma. Well done.
Yeah, this was from my previous experience because I also follow the production, so I take this opportunity.
Absolutely, it's definitely a good field to be into Six Sigma and the efficiency and how a process can be easily improved by implementing or not implementing steps and stuff like that. But you've got these very good professional involvement. So you've got quite a great rounded experience in this industry, I see.
Yeah.
Okay, well, moving on to myself, you're all familiar with who I am. I've been doing these webinars, and you can know where to contact me. I wanted just to say we do have our usual QR codes for LinkedIn, and especially for our Knowledge Center, because there's a lot of great information there. And we do have a technical paper based on this webinar today, which covers some of the topics that we will go over. And so I'm going to hand it over to you, Elena. You can go through the agenda with us, and I'm going to go off camera now, so I'll let you take over.
Okay, thank you, Jason. With me, there are three colleagues of mine. First of all, we have Cinzia Pagani, that is our co-founder and vice president of AM Instruments since AM is born. Then we have Annalisa Soldi, that is the project manager of all the contamination control solution products that we have in AM. And Andrea Weiss, that is our manager in the export team. So she's covering all the export customers that we have.
Wow, you brought a big team with you.
Yeah.
I think that just goes to show the subject matter that we're discussing today is very important, and how in-depth AM Instruments has invested in professionals to cover these type of products and serve the market.
Okay, so our agenda will include a brief introduction of AM Instruments. Then, of course, we will see something related to the GMP Annex 1, the new one. What is Pharmaclean in AM? The relation between Pharmaclean and Lighthouse Solutions Air Samplers. What solution does Pharmaclean provide to our customer? And of course, there will be also a section for questions and answers.
Right, looks like an interesting agenda that we're going to cover.
So let's start. AM is an Italian company that was founded 35 years ago in 1990, and is headquartered in Bate, in Italy, that is between, let's say, Monza and Milan. We are specialized in the contamination control solution, in particular for clean rooms, but we have also customers related to laboratories, controlled environments in general. And this company is today structured in three main divisions. We have AM production, which focuses on developing contamination control technologies. Then we have AM distribution, which offers a high quality products from also thanks to our international partners. And the third one is a AM service, which provides GMP services like, for example, biodecontamination, validation, repackaging, training, and calibration of instruments. And we operate under our four key, let's say, values that are critical thinking, transparency, emotional intelligence, and audacity. We are also certified ISO 1001, and a term that we create that is GMP consistent, and later on we will see what this means. We have also developed a specialized brand called Pharmaclean, which focuses on decontaminated packaging and material transfer solution, and AM Tech, which instead delivers advanced contamination control technologies. In the next slide, I want to, let's say, have an overview of AM through the year. So we said that AM is born in 1990. In 2008 begins the collaboration between AM Instruments and Lighthouse. Then in 2017, the new Pharmaclean production covers and sterility protection solution. So our first site of production was inaugurated, and the year later starts the production. 4 years later, so in 2021, there was the starting of the second site of production related to sterilization and packaging solutions. And what about today? Today, IAM consists of 170 people we have more than 3,000 products and approximately 880 active customers worldwide. We have 24 trademarks and 4 patented technologies. And in the next 3 slides, I like to make an overview of the 3 different divisions that I have just mentioned before. So the first one is AM production. This section groups all the products and technologies through thought to ensure optimal performance and unparalleled safety into clean room environments. As you can see in the pictures, there are some examples, and this section includes a packaging and sterilization solution, here represented by a cover made in Tyvek, and of course, biodecontamination system. Not presented in the pictures, but however, in our portfolio, we have also cleaning tools, accessories, clean room stationery, laminar flow, and disinfection end. Jumping to AM distribution. In this section, we work with international partners that ensure a selection of best-in-class quality products. And in this case, our portfolio consists of particle monitoring, air sampling, but also garments, detergents, disinfectants, maintenance tools, and many other products. And the third one, that is AM service, which includes GMP services like on-site biodecontamination, repackaging, process validation, control, and also our AM campus training. So to train people, in particular operator, in their operation.
Wow, I would just like to let's unpack all of that at the moment, because that's quite a portfolio of experience in clean rooms. A lot of obviously you're in production yourself, so you were understanding the production processes and the need to have a sterile environment for production, and then you have the products themselves, a wide area of different types of monitoring products and products for sterility and disinfection. So you cover a lot of the different segments within the clean room environment. So I guess you've built up a lot of expertise, good knowledge in the company to be able to provide from production to products and services. So that's special for a company to develop that amount of experience. That's probably the point that I wanted to make.
Yeah, we would like to be, let's say, partners of our customers. So to have the possibility of being, let's say, supplier all around, it's of course an advantage also for our customers if there is a good relationship between us and our customer.
I agree with you, because then within these different segments of the industry, then you start to connect the dots of everything in between. You know, the different, oh, this leads on to this process, and this is why this is important at that stage, and that's why we're monitoring here, that's why we're disinfecting down here. And you know, I think, do you also do clean room certification yourselves as well, certifying clean rooms?
Yeah.
Oh, so
Exactly.
you understand the whole process. That's great. Let's continue on.
Okay. So we have already talked about Annex 1, the new Annex 1. So I want to make an overview of the main topics that are inside the new Annex 1. First of all, the last revision has significantly expanded the guidance on the manufacture of sterile products. In detail, the requirements regarding clean room design and qualification, barrier technologies, environmental and process monitoring are becoming stricter with respect to the previous version. And now the Annex 1 does not cover just the finished product, but also APIs, excipients, packaging. So let's say it's become more global regarding the aseptic processes. So to summarize, the revision aims to ensure a risk-based approach to sterile manufacturing with greater focus on contamination prevention, process control, and documentations. if we want to make a very quick summary of the new Annex 1.
The last thing that you have underlined there, greater focus on contamination prevention. And that's the topic that we're going to be really discussing today, with the use of the sterility covers in that process.
Yeah, that's why I wanted to underline some points, because are the one related to what we will see in the next slides. Exactly here in this slide, I wanted to go a little bit inside the Annex 1. Here I have reported some paragraphs that are focusing on the topics that we will see on the presentation. So the first paragraph, the 8.8, contains a sentence that say, the presence of materials liable to generate particles and fibers should be minimized in clean rooms. So the first hint that Annex 1 gives us is that we have to reduce the possibility that the packaging add the particles. Then we have the 8.56 that say that the items to be sterilized should be packaged in a protective barrier system, which allows removal of air and penetration of steam and prevents recontamination after sterilization. So here we can understand the importance of the permeability of the packaging on one side, but also the fact that the packaging should be a barrier, because should prevent the recontamination if there is also in particular a period of storage after the autoclave cycle. And this point links to the last paragraph, the 8.46, that says that if sterilized items are not used immediately after sterilization, these should be stored using appropriate sealed packaging, and maximum hold time should be established. And we will see that all these characteristics are fulfilled by Tyvek. So I wanted to make some, let's say, important words that could summarize what we have just seen about Annex 1. So the important words are minimize particle generation, the packaging as a barrier, the possibility to have the good penetration of steam, and prevent recontamination. And so now, how a material could fulfill all these requirements? Let's see how Tyvek is the best choice. So Tyvek is a 100% synthetic material made from high-density spellbound polyethylene. So it's constituted of continuous fibers. And this structure guarantees a microbial resistance. So we have the first answer, permeability. Microbial resistance is a barrier. Then Tyvek is an hydrophobic material, but has a porous structure that guarantees the vapor to penetrate, avoiding water, liquids, or also solid to pass through. So we have a barrier. It is a highly resistant material to tearing and punctures, and the packaging made of Tyvek are very easy to be removed and do not require the use of adhesive tape. It's able to reduce the operating time and the related risk of contamination, both in packaging and unpackaging phase. So it also answers to the requirements of prevent recontamination of the items. In this way, we arrive to our Pharmaclean that we have already mentioned, but what is Pharmaclean? Is AM business unit specialized in the creation of clean room contamination control products and technologies. Today, it's constituted by 2 clean rooms that are in grade C operational, so there is a constant counting of particles in the air. And the first line that we call the Pharmaclean 1 is in this one, is dedicated to the production of packaging made in the so-called soft Tyvek, so in a wooden-like material, with which we produce covers and bags typically. And the second line is dedicated instead to the production of packaging made by Tyvek and the PET PP that are used to build bags and reels. And we have let's say something very particular in our line, because integrated to our line, we have a system, a proprietary system called Safe for Clean, that aims to the decontamination, the so important decontamination of raw material surfaces, this way reducing the total particle load. Of course, we have tested the efficiency of this system, and to do that, we have performed a bioburden test and liquid particle counting test on the raw materials, so on the Tyvek and the PET-PP, before and after the decontamination treatment itself. And here below, I've reported some test results that we obtained. So, for example, considering particles greater than 5 micrometer, we have a reduction up to the 60% on Tyvek and 40% on PET-PP. After this process, we start producing the final products in Tyvek. How to choose the proper solution for the different items? On one side, we have the soft Tyvek for covers and bags. I have put a photo on the left represent a mini cover. They are ideal for primary packaging. They are very used to contain machine parts or to cover, for example, stopper ball or in general surfaces. And they are very practical, so are very easy to be removed in narrow spaces like, for example, isolator. On the other hand, we have the Tyvek PET-PP bags and reels that are very useful to protect let's say the good outcome of the autoclave cycle when we have items with sharp edges or that are very heavy. It guarantees a high strength and has a superior microbiological barrier. The bags that are represented in the picture on the right are provided with a thumb notch and are peelable, thus helping the operator in performing a very slow opening of the packaging, thus preventing even more the possibility to have generation of particles. And the PET-PP transparent film guarantees the visibility that is particularly useful when the customers have a lot of tools that have a similar shape. In this way, it is possible to distinguish them. So we arrive to the kit.
Yeah. What we have here then is just an overview, folks, of the type of the air sampling equipment that we have. This is just some of the sampling heads that we start off with a washdown cover. Then you have the sampling head, the impactor base, and the Petri dish holder at the end.
We call it the removable upper impactor base.
Good terminology. I call it a media holder. Same thing, but it's also great because it's magnetic as well. So you don't need to use tools which will generate particles in the clean room. So just examples of the primary packaging and the secondary packaging solutions as the product is transported to the sterility cycle in the autoclave and then packaged afterwards and ready to be used in the next batch of product that's been developed or manufactured in the aseptic process. So it's we cover all the different sizes that we have. I just wanted to show that. You have a video here as well. You want to talk your way through that, Elena?
Yeah, in this video we show how to proceed with the primary packaging. So we have two covers for the head and the base of the impactor. And you can see how easily and quickly the operator is able, of course, is speed up, but it's very easy. Then we have bags for the two other items. Of course, here are not welded, but the bags should be welded. And then as secondary package, we have 4 bags, all of the same dimension, that will be provided with a hole to be hanged up in the isolator. And you see how it seems how it is repeatable, the procedure of the packaging.
Absolutely, it does look like a very easy process to follow. And before we get into the advantages, there's also a part in Annex 1, which is section 8.2, and I'm just going to read it off. It says that primary packaging containers and components should be cleaned using a validated process. And it says this applies to sampling equipment that comes into contact with the sterile process. Can you talk a little bit about the validated process and what validation? I know you covered on some of the validation you've done internally, but in terms of I'm a new customer and I want to use this product, how do I go about validating that process for myself internally, and what support do I get from AM Instruments on that?
Of course, the validation depends on the SOP and on the production that the customer has. We have performed our own, as we have performed tests autoclaving our packaging. And we have used a specific autoclave with parameters set for our autoclave cycle. We have the results, we can provide them, and we can support the customers with their validation process of the autoclave and of the validation of the autoclave. That, of course, if a customer arrives from a different packaging material, the autoclave cycle has to be revalidated for sure. And we can support them with our documentation from our quality assurance department.
Any other type of support that you would offer as maybe subject matter experts or that would help in a project to offer your consultancy service, or is there stuff like that?
Yeah, typically, if there is the chance at the beginning of the project, well, of course, it's much more easily if I can go on site and see how the pieces are taken off, but especially remounted on the line, because of the airflow and all this kind of stuff that should be taken into consideration. And also to see the autoclave load could be useful. And then we provide the recommendation that also Annex 1 said, so how to put the different items in the load. So if possible, with an angle of 45 degrees, how many packaging they have to consider. So the number of, let's say, passages of classes correspond to the number of packaging, a primary and a secondary packaging. And so this from a, let's say, more practical point of view. But of course, for the validation of the cycle, we have documentation to support them.
That's great. It sounds like you do have a lot of support there to help customers who are going to look into this technology as a barrier to help with the contamination control. Do you want to talk about some of the advantages of Tyvek?
Yes, of course. Let's say in particular, why one of your customers, Jason, should consider our Tyvek. First of all, because we have specifically studied it for your Lighthouse Air Samplers kit, because our Tyvek solutions are 100% compliant with Annex 1. And because you will put a sample of our kit together with your air samplers kit, so the customer could test it.
Yeah, we're going to be sending out a sample kit to anyone that purchases a new product to have a look at the product, to look at the Pharmaclean, the process, and see if you want to implement that into your cleaning solution, particularly transferring the sampling heads and the holders and everything to and from the autoclave.
And then I put some dots regarding more in detail why it is compliant to Annex 1, because as we have said, Tyvek is an excellent microbiological barrier for its nature as a low particle release for the structure of its fiber. Because Tyvek solution has also, we have seen from the video, guarantee a reduction of the operating time that is linked to a repeatable process that is also so important. And moreover, you don't need for the packaging to use adhesive tape, for the unpackaging to use scissors that could, both tape and scissor, cause particle release.
Particle generation, yes, exactly.
Exactly.
Moving on, I just want to show this is some of the portable products that we have, the active count air sampler. Your Pharmaclean will work with the active counts and also our remote active counts products as well, just to show some of the devices, the sampling devices and the products, the sampling heads, the holder, and the impactor base, which can all be autoclavable. And the Pharmaclean solution works for all of that. Okay, I guess that brings us into question time. And if you like, we can introduce some of your guests, your colleagues who can help with some of the questions. I guess the first question is like, overall, what is the advantages of using Pharmaclean? Now, we discussed the contamination process or trying to eliminate contamination during the transfer process. The standards themselves, because this is a 2022, this came out, so it's a requirement. And we discussed how it can be implemented. So yeah, I'll let you dig a bit deeper into the overall advantages of Pharmaclean.
Yeah, this is a good question to summarize what we have seen all over the presentation. So let's say first, I will say that in particular, our kit is thought for your air sample kit. So for example, we have decided together to put this hole in the secondary package so the bag could be hung up in the isolator. So it should perfectly fit the needs of your customer. And also because the base material, the raw materials, are biodecontaminated. This is also one of the main advantages, in my opinion, of our products. Also, last but not least, what we have just discussed, so the possibility to have a full documentation support for our customers.
It's extremely important to have that level of support and the right subject matter experts. That's why I spent a little bit of time when I was looking at your career, the company itself, all the different divisions and what you cover. So you're in a lot of different segments of the pharmaceutical manufacturing process, from raw materials coming into the warehouse to a finished product, injectable, vaccine, whatever, coming out the other end. You guys are in all of the different areas, as I can see. So I guess another question we have is with Pharmaclean, how will it enhance the contamination control strategy?
So AM manufacturers at the same level of quality that our customers are required to obtain. So this means that our products embed the answers on the managing of the packaging in aseptic processes. I think that this is all, and we have to thanks a lot our quality department, where there are people coming from the pharmaceutical industries, and the manager is an auditor herself. So
Yeah.
this helps AM a lot in obtaining this high level of quality that we can count on.
Not only that, and I guess the design of the product will fit into what the industry is expecting. But you know, Annex 1 does talk about the contamination control strategy for the whole facility. Well, let's move on then to our third questions. This is one. Do you think regulatory inspectors will be looking at these innovative solutions like Pharmaclean to address the Annex 1 update? What are you seeing from the field? What feedback are you getting?
What we see is that when we participate to events where inspectors are there too, there is indeed a growing attention to particulate contamination, which is especially critical to patients because fibers are pyrogen. And as this issue cannot be mitigated through sterilization, and the raw material is not produced in a controlled environment, the only solution is to reduce the initial burden, and this is what we achieve through our safer clean station. Of course, there is Andrea Weiss that she has much more experience than me on the field and PDA and this kind of very technical
I'd love to hear what Andrea has to say.
Nice to meet you. Nice to join the discussion. Thank you.
Welcome, Andrea.
Well, I can confirm what Elena already has mentioned. As I'm attending most of PDA ISPE meetings around in Europe, the current question or the current topic that inspectors bring to the discussion is always about particulate contamination. as this is, let's say, particularly tricky because not as easy mitigable like microbial decontamination by sterilization, because a particle, even if sterile, still remains a particle. So if you don't start from scrap, looking at a very early stage at the raw materials that you're bringing in, when I always tell my customers, look, pulling things through a clean room doesn't make them worse, but for sure it doesn't make them better. So you have to actively contribute and lower the burden by activities that you consider useful for getting a better process. And Annex 1 is very clear, as already Elena has already evidenced before. And fibers, let's say very easy, are the last thing a compromised patient needs. Because if the patient then gets fever, it's for sure not the expected result of an injection. And as we are very patient-centric before doing any else, let's say, reasoning, first of all, the patient's wealth, then we have to act in this way. We have no choice.
Absolutely. I did mention that Elena mentioned in the introduction, empathy was one of their core values, and it's definitely we definitely need more of that in the industry, because we're dealing with people that have compromised immune systems. So it's in all of our best interests to implement whatever controls we can put in place to minimize that contamination, which kind of brings me back to section 8.8 of the Annex 1, which says precautions to minimize microbial, endotoxin, pyrogenic, and particle contamination should be taken per site's contamination control strategy during the preparation of the aseptic process stages. So it's really telling us, we need to implement more strategies to minimize the contamination for the safety of the end user, these patients with compromised immune systems. Yeah,
for sure. And always look at the whole picture and the focus, which is very clearly the patient behind, not the sterile process, but the people that need to benefit from the process. So we really have to think it in 360 degrees.
Yeah, definitely. Before we wrap up, this kind of leads on to like a question that any customer watching this webinar would have, was like, how easy would it be to implement the Pharmaclean covers into the autoclave process for their application? Maybe if anybody wants to jump in and answer the question, because we kind of showed the validation process, but yourselves as AM Instruments with other Pharmaclean products, how easy is that for your customers to implement these products into their processes?
So let's say, Jason, in terms of autoclave validation, as I have already told, if the customers arrives from a different material or no material for packaging, of course, the cycle should be validated or revalidated. Instead, from the operator's point of view, typically they, as what I saw in the field, they are very grateful because they are very easy to use. The primary packaging, it's easy to be removed inside the isolator, for example. So in particular, the attention on the unpackaging and the reassemble of the machine parts in the isolator is the challenging part. And I think that with our solution, we try to help the operator in this sense.
Yeah, definitely. And it seems like you've gone through this process with many other customers in helping them from establishing this product into their process. So I did have something to say, and as I was talking, it just slipped out of my head. Maybe I'll come back in again. But just in closing, I'd like just to have a quick discussion of the product itself, and are you seeing feedback from the field, especially, I guess, whenever there is a CFU event established within a septic environment, there's a root cause analysis has to be investigated. I think one of the big advantages of implementing this product is that you're going to put the focus away from the cleaning process, more so because you've put these extra steps into eliminating the particular contamination at that stage, whereas the root cause analysis and the investigation could focus on other areas of the process first before looking at the Pharmaclean process, because we've basically added that extra assurance into the process. I wouldn't start looking there. I'd look somewhere in the weaker links of the actual process to see where did this CFU come about. Obviously, the location is going to be a big giveaway to start your investigation. But in terms of how you've seen the uptake in industry for this particular product?
One of the best results that a customer told me was that they were able to reduce the garments of their operator, because now the risk of having a contamination from the operator, that is one of the most possible one and critical one, was reduced because of the movements that the operators do while unpackaging the items. And I think that this is a very good outcome.
So straight away, they saw that weak link and they were able to see that there was contamination coming from the operators before they used the sterility covers.
Exactly. One customer point out this to me, and also, of course, a reduction in the operations. Apart from everything concerning the sterilization, the particle release, that is something, let's say, fixed. We know that, but these are the accessory benefits that we saw on the field. And yes, when I have the chance and I go for the first consultancy on place, I always ask for operators if they are not there, because they helps me a lot in finding out the appropriate solutions, because I may think that something could work, but they have the correct view of the process, and so they are the best people that could help me in finding the solutions they need.
Yeah, exactly. And just in summary, I'd like to ask if Andrea or Cynthia, if they'd like to summarize the product or jump in and close out the webinar today for us and talk a bit more, some more success stories with this product or anything like that.
Yeah, I wanted to add something with regards to the packaging. I mean, packaging is always just very strictly concerning one moment of the activity, which is packing. Okay. What is really important and game-changing or changing the situation mostly is the opening, the reopening, the unpackaging, what has Elena mentioned before, and where most of the contaminations, I would say, can happen, because if you have a high particle load and you intensively move by opening, peeling or cutting with scissors or whatsoever the packaging, and then you move it around for getting out the objects, this is really the critical moment. And if you do not have a properly decontaminated material at hands, you may get decently over the packing moment, but the unpacking then is really getting critical because you're moving too many things. So apart from the easiness of putting things where they need to be covered and then in the packaging, also the last moment, the unpacking of goods, is really, really important, and where you see the difference between a particle loaded packaging or a decontaminated packaging. And I mean, Jason, this is something that I don't have to teach you anything with the measuring of contamination. You know what we are speaking about. So this is where customers are mostly happy having really material with the fewest burden available. And what we nowadays can offer by the Safe for Clean station, which is our unique sales proposition, is that we really can offer the state-of-the-art situation, the best condition the packaging raw material can get into with the lowest burden available. And I think having this premium starting point is really the best condition for getting a premium packaging.
It sounds like you definitely eliminate a lot of the weak links in the process. I want to say a big thank you to AM Instruments, who are a special partner. And thank you, Elena, for your overview of the Pharmaclean process of AM Instruments as a company and the wide range of services and products that you offer to the clean room industry and your own experience as well. And also thank you, Andrea and Cynthia, for joining us and offering your overview, Andrea, of the environment and some of the feedback on the implementation of this particular solution. It is a solution that we're offering here for the update in Annex 1. So thank you very much. And just in closing, unless you guys want to have anything else to say before we wrap up?
It's always a pleasure thanking you for having us. And, most of our job, let's say, is being a consultant, because we have a broad view on contamination control and on the aseptic process. And you're always welcome with any kind of questions.
That's great. Thank you very much, Andrea. And thank you, Elena, for your great overview.
Thank you, Jason, for the opportunity. Thank you very much.
Okay. Well, folks, thank you for your attention today. And before we wrap up and close it out, I just want to show those links again for the Knowledge Center and also AM Instruments. If you get onto AM Instruments website, there's more information about the company, about the Pharmaclean products, and you can get a lot more details there. So if you want to get some more knowledge and then grab that technical paper, you can go to the Knowledge Center and you can download that paper from there. So thank you very much, guys, and I look forward to you all joining us in next month's webinar. And again, special thanks to our colleagues at AM Instruments for their great overview of this Pharmaclean product. And I look forward to catching up again. See you all next month. Bye-bye.